Welcome to your YouTube channel NCERT e-teaching decoding NCERT. I am Dr. Kailash Mina going to explain the mechanism of DNA replication, the machinery and the enzymes from class 12th biology, chapter 6, molecular basis of inheritance. In my previous videos I have discussed about the structure of DNA, the experiments for the search of genetic material and the proof of the DNA is the genetic material. Next, we have discussed packaging of DNA in chromosomes in a eukaryotic cell, and in my last video I have explained the experimental proof of semi-conservative mode of DNA replication. So in that video we learned that how in 1958 Matthew Meselson and Franklin Stoll gave the experimental proof of semi-conservative mode of DNA replication. So in that video here is a brief summary that three type of mode of DNA replication were possible: semi-conservative, conservative and dispersive. And out of these three, semi-conservative, conservative and dispersive, Meselson and Stoll proved that semi-conservative mode of DNA replication is the mode of DNA replication which actually happens within the cells. So for learning more about that experiments, you can watch my previous video of the same topic. Now coming back to the current topic, so in this video we are going to learn about the mechanism of replication, and after finishing this video we will be able to know why replication of DNA is semi-conservative. Next we will able to know about the enzymes and proteins involved in DNA replication, and we will also be able to learn about the differences between DNA replication in eukaryotes as well as in prokaryotes. So for achieving these learning outcomes from this topic or from this section, we need some previous knowledge. As in my old videos related to chapter 6 or in previous chapters, I always mention what previous knowledge we need to understand that topic. So first thing, we need the understanding of structure of DNA, which is very crucial if we want to learn about the DNA replication or copying of DNA, because the copying of DNA is dependent on these structural details. Here two strands of DNA which are antiparallel, and thymine is base pairing with adenine, and in the same way guanine is base pairing with the cytosine. You can learn more about this base pairing in my previous video, which is about the structure of DNA. And one interesting fact is there that when Watson and Crick in 1953 they gave the structure of DNA as the double helix, they also pointed out this fact that from this structure of DNA which they gave, one immediate copying mechanism is suggesting. So they also pointed out that mechanism which we are going to discuss here, but at that time there was no experimental proof available. Now we know the experimental proof was given by Meselson and Stoll in 1958. The next thing we need is the differences between prokaryotic and eukaryotic cell. Let me tell you very frankly that from class 9th onwards, if you want to study anything in biology, you must be aware about the structure of prokaryotic and eukaryotic cell. And for this chapter, the molecular mechanisms which we are going to discuss, every mechanism we are going to relate with prokaryotic cells and eukaryotic cells. And finally, the third thing which is required to understand the mechanism is the experimental proof, which already I have discussed in my previous video. Now coming back to the current topic, which is machinery and enzymes. Now let's consider this diagram. So as from the experimental proof, we all know that the replication of DNA is semi-conservative. It means the previous DNA molecule, which is denoted here with blue color, this DNA molecule will unwind or denature, or it simply means that both of the strands will be separate by breaking hydrogen bonds, not covalent bonds, because this blue line represents the sugar phosphate backbone. It means the deoxyribose and deoxyribose and phosphate backbone, which is represented by blue line, and they are joined together by covalent bonds, and the units are of course called nucleotides, which we have discussed in our structure of DNA video. And this DNA unwinds here by breaking the hydrogen bonds, and when this unwinds, this very simple diagram which is showing unwinding of DNA, we can spot the fork like structure, and that is why this portion is called the DNA replication fork. So when we study the sequence of nucleotides in DNA or in RNA, or if we are studying any process, whether it is DNA replication or it is transcription or it is translation, or simply we are going to read any sequence in our theoretical exams or any question, always we must remember that we are going to follow the sequence from 5 prime direction to 3 prime direction. I have discussed this 5 prime direction to 3 prime direction in my previous videos of DNA structure. You can see those videos if you want to learn about 5, what is 5 prime, what is 3 prime. Here briefly I am telling you that 5 prime and 3 prime are the carbon number 5 and 3 of pentose sugar in the structure of DNA or in the structure of nucleotides. So here 5 prime means 5 number carbon and 3 prime means 3 number carbon of pentose sugar, and they are denoted at the end. Why they are in the end? Because the end, the final nucleotide have its 3 number carbon which is joined with OH group, 3 prime OH group free, and this 5 prime carbon with phosphate group is free, and in between all nucleotides the all carbons of sugar are busy. So very briefly, here every DNA sequence, every RNA sequence have one polarity which is 5 prime to 3 prime direction. So we must keep in mind that if DNA is replicating, if DNA is making its copies, if DNA is making RNA, if RNA is processing, or RNA is making another RNA, or RNA is making another DNA, or RNA is making another protein, in any molecular mechanism we will read any sequence of DNA or RNA from 5 prime direction to 3 prime direction. Even in biotechnology experiments, which we are going to learn in unit number 4 of our class 12th biology, the biotechnology, every sequence for every numerical, any question, we will always read from 5 prime to 3 prime direction, because our cells are also reading in the same way. So 5 prime to 3 prime direction you keep in mind, and this direction is going to explain this diagram also. Now you can imagine that blue color lines are representing the old DNA molecule, and red color lines, whether it is continuous or discontinuous, it is representing the new DNA molecule. Now what is happening? The DNA is unwinding and the new DNA is forming, and this new DNA is forming in the direction from 5 prime to 3 prime. Now if old DNA is unwinding or the hydrogen bonds are breaking in the old DNA, what we get? We get two poles or two ends of old DNA, which is denoted as 3 prime and 5 prime. Now as we know from the structure of DNA that the two strands are anti-parallel, it means when new DNA synthesis happens, what happens is that in front of 3 prime the new strand will form 5 prime, and we know that always DNA synthesis will happen from 5 prime to 3 prime. So in this strand it will go like this, and in the next strand it will go like this from 5 prime to 3 prime. So let me explain this structure by giving you a rough idea how it happens. Wait for a minute, I have arranged some rough sketches for this presentation. Now here you can see that the old DNA molecule is represented by here with slightly blue color, and if it unwinds, here is the replication fork, and then new DNA synthesis begins from 5' direction to 3' direction in this strand, and from 5' to 3' direction in this strand. Let me have a very fresh clean screen to show this mechanism to you. Now imagine that we have this blue color old strand, it is unwinding to make for making replication fork, and it is the old DNA molecule. Now it is 3' and this is 5' and this is 5' and this is 3'. Now this end is joined together with hydrogen bonds, and this has been unwinded for the replication of DNA. Now what happens, that new DNA forms in from 5' direction to 3' direction here, and 5' direction to 3' direction here, because we know that DNA replication will go always from 5' direction to 3' direction. And because of this limitation, what happens if cell or DNA wants to continue the DNA replication, what it can do, it will unwind again, because the whole DNA, like you can see in this diagram, you can see that one bacterial DNA molecule is shown here, and it is unwinding gradually. You can see the gradual opening here. So DNA is unwinding gradually, then DNA replication begins, and we are taking only this part as replication fork or this part as replication fork for ease of study. So here you can notice one more thing which I am going to explain later, that prokaryotic cells have circular DNA molecules which have single origin of replication, and eukaryotic cells have a linear DNA molecule which have multiple origin of replication. Now coming to the current slide, what happens here is that, as we have learnt in with this diagram, the DNA is open up making replication fork. DNA replication happens from 5' to 3' direction in both of the ends. But what happens next, it is more interesting. What happens is that, let's imagine this was the previous strand and it was unwinded like this, and new DNA was forming like this. Then now it will unwind more, this DNA will unwind more for making more new DNA. So it will unwind like this. We have old DNA like this. Now we have this new portion for starting new DNA. Now what happens is that, because as the polarity goes like this, 5' 3', 3', 5' for the old DNA strand, and from 5' to 3' for the new DNA strand, what happens when cell synthesizes or DNA synthesizes new DNA strand, it will go like this continuously here. But what will happen here, because DNA can go always 5' to 3' direction. Now what about this part, how can be DNA formed in this way? So again it starts anew, to make a new DNA molecule from 5' direction to 3' direction, and this is why every time it unwinds new this DNA molecule, every time cell have to start again from 5' to 3', from 5' to 3'. So this happens, DNA keeps unwinding and unwinding, and then new DNA strands forms like this. At one strand it is continuous, and in the second strand it will be discontinuous. So coming back to the previous slide, now I hope you are able to understand this diagram from NCERT, why this replication fork goes in one end it is continuous, in another end it is discontinuous, because of that 5 prime to 3 prime direction problem and the unwinding problem of DNA also, because it is a short part of DNA. It is not complete DNA, whether it is in eukaryotes, whether it is in prokaryotes. DNA will unwind slowly and gradually, and then new DNA synthesis will happen from 5 prime to 3 prime direction. Both of the strands are antiparallel, and again the new strand which is synthesizing is also antiparallel. So because of this problem, what happens, we got discontinuous synthesis at one strand and continuous synthesis at the another strand. Let me give you one more name for these strands. We will call this strand continuous is leading strand, and this discontinuous new strand is called lagging strand. You can also correlate with this lag and log. What do we mean by lagging strand? Because it is discontinuous. Leading strand, it is leading continuously. Now do not bother about the details. I have copied all the details as it is from NCERT for the correlation point of view. Now, so we know now we know about the replication fork, the continuous and discontinuous synthesis of DNA, and the 5 prime to 3 prime anti-parallel direction. And we will always read DNA sequences from 5 prime to 3 prime direction. DNA will always synthesize from 5 prime to 3 prime direction, and RNA and protein synthesis will also happen from 5 prime to 3 prime direction. So this is the guru mantra of the molecular biology, that you always read nucleotides or in any sequence from 5 prime direction to 3 prime direction. Now again, coming back to this diagram, this beautiful diagram is showing the differences between prokaryotic cells and eukaryotic cells. As we all know that prokaryotic cells or bacterial cells have a single DNA molecules, and we eukaryotes including humans have many DNA molecules, and those molecules are called as chromosomes, and those are linear DNA molecules. And in case of bacterial cells, the DNA is circular. You can easily see in the diagram. So this circular DNA, whether it is circular DNA or it is linear DNA, it will unwind first, then there will be a space which is called replication bubble, there will be replication fork, and there will be continuous and discontinuous synthesis. It will go on, and finally the complete DNA will be duplicated or replicated. But yes, this process will take time, because we humans have 3 billion nucleotides in 23 chromosomes, and if you take all 46 chromosomes for one cell altogether, it will be 6 billion nucleotides. So it will take time. So what happens is that prokaryotic cells have single origin of replication and eukaryotic cells have multiple origin of replication. So what is origin of replication? As the name indicates, the DNA replication cannot start randomly from any place in DNA. It will start from a definite place, which is called origin of replication. In case of bacteria or in case of E. coli, it is ORIC. It is called ORI, origin of replication. And in case of eukaryotic cells, you can see here that we eukaryotes have multiple origins, and because of that we are also able to duplicate our chromosomes or to replicate our DNA molecule in the time limit. So this idea I wanted to convey to you. Finally, the mechanism of DNA replication. So DNA replication happens with the help of many proteins, and many of those proteins are enzymes. So here I am giving you very simple idea as from our previous discussions in the structure of DNA, and as we have learnt in class 11th biology biomolecules, we know that DNA is a polymer of nucleotides. What is a polymer? Polymer is a chain of nucleotides joined together by phosphodiester bonds. So we can simply assume that if we are talking about making a new DNA molecule, we are talking about new polymer which will form by joining together nucleotides with phosphodiester bonds. So simply we need one enzyme which can join together these nucleotides into a complete polynucleotide chain, and that enzyme will be called polymerase, because it is doing polymerization. So if it is making DNA, it will be called DNA polymerase, and or if it will make RNA molecule, it will be called as RNA polymerase. So we will learn about RNA polymerase in the next topic, which is transcription. So here this enzyme is DNA polymerase, which is main replicative enzyme. But what is the problem with DNA polymerase is that it cannot synthesize or cannot start DNA replication from scratch. It will need a lot of assistance from other enzymes and proteins. So what was those assistance? It will be very interesting to know, and it is very fun for knowing that if you have a DNA molecule, first you need to unwind it for making a new DNA molecule. What is unwinding? Unwinding is simply denaturation of DNA or breaking the hydrogen bonds. So we need one enzyme which is called DNA helicase. Here it is shown, DNA helicase enzyme for unwinding of DNA. And if you unwind any thread, if you unwind any rope, if you have dealt with ropes, you know that we find some twist if you unwind or rewind anything. So for removal of those twists, we have one more enzyme which is called gyrase or topoisomerase, which is not discussed here. It is discussed in our NCERT textbook. So we first need to unwind the DNA, and who will do this job? This job will be performed by DNA helicase. After unwinding, rewinding may happen. As I have discussed in my previous videos, the denaturation of DNA and renaturation of DNA simply means breaking the hydrogen bonds and rejoining the hydrogen bonds according to their complementary base pairing into strands. So denaturation is also possible and renaturation is also possible. If you boil the DNA molecule at 100 degrees centigrade in the water, in the buffer, it will get denature, and if you cool it down, it will again renature. So denaturation of DNA is a reversible process. So for because why we are unwinding DNA, why we are breaking the hydrogen bonds? Cell is doing this for making new DNA molecule. And if you unwind DNA, and if it will be rewinding, because we know that the temperature of cell is constant for human beings, it is 37 degrees centigrade. We are not boiling DNA, we are not cooling it down. So if we are unwinding DNA, there is a possibility of rewinding of DNA. So to avoid that possibility, there are single strand binding proteins. These four marbles like structure are showing here to show the single strand binding proteins. So again, the next thing is, first was DNA helicase, which was unwinding the DNA, and the next one is single strand binding proteins. These proteins will hold the DNA strand for making new DNA molecule. Again, DNA polymerase, even after this, it cannot synthesize new DNA molecule. It need a short RNA sequence which is called RNA primer, which is also called primer, from 5' direction to 3' direction. So primase is a kind of RNA polymerase which will perform this job. So this is very complicated process. So first DNA will unwind, DNA helicase will do that. Next, single strand binding proteins will hold the single strand of DNA. Then third, the primase will synthesize a short RNA sequence. Then now DNA polymerase will come and start synthesizing DNA molecule. So here you can locate the primers synthesized by primase. Now here you can correlate, this is the leading strand, this is the lagging strand. So here you are noticing two primers. So every time you know about the problem from 5' to 3' from my previous slide, then we every time DNA get unwinds, we need new molecule synthesizing in the lagging strand. So every time you will encounter a RNA primer. So DNA polymerase again you will encounter the words DNA polymerase 1, DNA polymerase 2, DNA polymerase 3 type of enzymes. So let me explain. In bacteria, we find so many types of DNA polymerase: DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, DNA polymerase 4, DNA polymerase 5. So these sequences were given from the sequence of discovery. So we discovered first DNA polymerase 1, and then we discovered DNA polymerase 2, then we discovered DNA polymerase 3. But the main replicative enzyme in E. coli or in bacteria is DNA polymerase 3. So here, when I say the DNA polymerase will continue the DNA synthesis, we are talking about DNA polymerase 3. Although DNA polymerase 1 also can perform this task, but it is not that much of efficient. It will come later. It will perform another task in DNA replication. So this is how DNA replication will happen in very brief. Although it is a very detailed process, but this much of detail is not included in our syllabus. So here we have one idea of machinery of the replication. So let me briefly introduce the functions. The first one was the DNA helicase separates double stranded DNA into single strands. Next was single stranded binding proteins, binds to single stranded DNA and prevents it from reforming a double helix. DNA primase, again the third one, synthesizes short RNA primers, again DNA polymerase, synthesizes DNA in the leading and lagging strands, removes the RNA primers, filling the gaps with more DNA and proofreads newly made DNA. So there are so many functions. I have not discussed these. So let me discuss this first. So what happens is that when you complete the DNA synthesis, when cell completes the DNA synthesis, these primers remains between the DNA fragments on the lagging strand or also one RNA primer in the leading strand. So who will remove these primers? Here DNA polymerase 1 comes. Previously I was saying that DNA polymerase 3 was doing the job. DNA polymerase 1 remove these primers. Again, if you remove the primers, there will be gaps. So who will fill the gaps? Who will make new DNA molecules in those gaps? DNA polymerase 1 itself. So DNA polymerase 1 will remove the primers, DNA polymerase 1 will fill the gaps. Again, if you fill all the gaps, there will be some cuts there. These are called nicks. Nick means a cut. So who will seal those nicks if there is any nick? Because if you are removing some segments, you are reforming some segment, there will be a few nicks, and these nicks means no phosphodiester bond between the sugar phosphate backbone. So DNA ligase is one more enzyme which fills these nicks or seals these nicks. So this table will give you the idea of this brief machinery, although there are many enzymes involved. As I said that topoisomerase or DNA gyrase for removal of the test, but this is not included in our syllabus. So these are the main replicative enzymes, and this diagram again shows the point of view of those fragments, and those fragments are called Okazaki fragments. Which fragments? One strand was continuous, another strand was discontinuous. We called continuous strand as leading strand, discontinuous strand as lagging strand, and these segments or fragments are called Okazaki fragments because of the scientists who discovered these fragments. So this is the brief introduction of machinery of DNA replication. Our next topic will be transcription, in which we will learn how DNA makes RNA molecules, and this transcription is the first step of gene expression. What is gene expression? Gene expression means we know from our chapter 5 and from chapter 6 that the sequence of DNA is called gene if it is making any functional RNA molecule or any functional polypeptide, or simply if it is expressing any trait from chapter number 5. So how DNA is able to express itself, how genes are able to express themselves, this mechanism is called gene expression, and it involves transcription and translation, and that is also called central dogma of molecular biology, which we have discussed in the structure of DNA also. And in my next video we will go in depth of these processes. So the central dogma of molecular biology involved the process of transcription and the process of translation. Here transcription means forming RNA from DNA, and translation means forming protein from RNA, and here in between there are many processes also which we are going to discuss. So thank you so much for your listening. In my next video we will learn about transcription.